To this point, we've been spending our time talking about how the immune system works and how the immune system's main role is to decipher between self and non-self, and then remove self as if it's, or remove non-self if it's threatening to the life of the individual. That then makes sense that we need to also make sure to discuss how organ transplants or tissue transplants can be viewed by the immune system as being, non-self. So chapter 15 is going to cover different aspects related to transplantation of both tissues and solid organs. And then we'll also look at what types of reactions can come about when these tissues or organs are rejected by the immune system. So there's three fundamental, ground level things that need to be considered when looking at transplantation. First, the transplant must be done in ways that the organs and tissues and the body, while it's going through the transplant, that normal function is maintained. So if an organ is transplanted from one individual to the recipient, the organ has to also be functional as it would in a normal situation in the recipient as well. This means that number two, both the health of the recipient and the transplant, the tissue or the organ, has to be maintained during surgery, and then the procedures to get the organ or the tissue to the recipient, so through transport as well. And then the third major consideration when thinking about transplantation is the immune system's response to that transplant. It's going to recognize it as non-self because there will be proteins that are non-self, and how can rejection of that tissue or of that organ be avoided due to the normal immune response. So those are kind of the three big things that we'll look at. And when we talk about rejection, it is everything to do with the external molecules on the surface of the cells within that tissue or within that organ. And these external molecules are the HLA, the human leukocyte antigens, but remember that HLA molecules are the same as MHC molecules. We just use the term in different contexts. So when we talk about antigen presenting cells and presenting and showing to T cells, we use the term MHC, major histocompatibility complex. Class 1, class 2. Those are also known as HLA, human leukocyte antigens. And it is the difference between these molecules that is recognized as non-self during any type of transplant. And so antigens, there's a few terms that go along with that. If you have antigens within the same species, so human to human, we call these alloantigens. Allo meaning different, but it's still within the same species. And then if these antigens provoke the production of antibodies, we call these alloreactions, or any type of response could be it. And then those are alloantibodies. So in a transplantation situation, there's actually two different types of alloreactions that can occur. One, the graft is rejected by the host's immune system. And so that's what's shown in this first image here, where you have an organ from an external individual being transplanted into the recipient. And then the recipient's immune system, which are these cells here, attack and destroy that organ or reject it. The other situation is when there is a bone marrow transplant or hematopoietic stem cell transplant, because in that case, the recipient's immune system is actually being wiped out and being reconstituted with new bone marrow. The new bone marrow has its own set of genetics and will make an immune system based on those genetics. And the potential with a bone marrow transplant is then that the hosts, or sorry, that the donor's now immune system can attack the recipients. And so we call that graft versus host disease. We'll look at that in more depth in the second lecture for this chapter. But those are the two major types of allo reactions. Oh, I guess I had the animations kind of off. Sorry about that. Okay, a couple terms before we get into our first type of transplant. So auto means same, allo means different, iso means exactly identical. So allo, then, if you put it in front of graft, that's going to be tissues transplanted from one site to another, or not allograft, autograft, from one site to another. We see this with like skin. Skin can be removed from one part of the body and used to like a skin graft and move to a different part. Shouldn't be any issue there because it's still self and the antigens are going to be the same. But that's an autograft. An isograft is if you have an identical twin, because the genetics are going to be identical between two individuals. They have the same genetics, and so to the body, it's going to look at like self. And so it is a transplant between two genetically identical individuals. Now, an allograft, allo is different. And so this is when a transplant is made between two individuals, same species, but different genetics. So they're histotyped. That means that they're going to be, what the next word is histocompatible. So they're alike enough that hopefully it won't initiate a huge immune response. And so a histocompatible donor is a donor that has a similar enough set to the recipient of those HLA molecules. So those are just some terms that we should bring out to begin with. Okay, so now let's dive into 3 different types of transplants. And the first one is blood transfusions. So this is the most common, by far, type of tissue that is transplanted from one individual to another. It is given to probably 1 in 4 people across their lifetime. And so that's really high incidence, right? And so this can come about for a variety of different reasons. Trauma being one we think of a lot, where there's maybe a car accident where there's a lot of blood loss or some sort of trauma like that. Childbirth is another very common situation where individuals will get a blood transfusion. And then if there are diseases where there's severe blood loss, there could be different anemias caused by a variety of different methods, or there could be maybe chronic blood loss through the intestines or something like that, where somebody becomes critically low, very anemic, and needs to get some red blood cells. So the donation, when you go and donate blood, and maybe you've done this before, but the machine that they hook you up to actually will take and separate out, and maybe you've donated plasma as well, but it separates out, maybe not the machine you're on, but if you're in a plasmapheresis machine, it will separate out the blood from the plasma. If you are just donating whole blood, there'll be a second processing that will separate out the red blood cells from the plasma. And so there's really 3 different, well, there's 4 different layers that the blood can be split into, but only 3 of them are utilized for transfusions. So first of all, the red blood cells, that makes up about half, a little less than half, depends on how high your hemoglobin or your hematocrit is. And when you take and you put it into a centrifuge, it'll spin down and the heavy stuff will go to the bottom and the lighter stuff will stay on top. And the red blood cells are what are actually donated if you get a blood transfusion. So shown here, those are packed red blood cells, and they're red blood cells that have been, plasma's been removed, the white blood cells have been removed, the platelets have been removed, and they are washed. So hopefully most of the antigenic properties are removed from those packed red blood cells. And that's what, so if you get a blood transfusion, that's what's actually being given. Plasma can be donated. There's no cells in plasma, and so there shouldn't really be any risk with a donation or with a recipient getting plasma. This would be, not having to do with anemia, but for other burn or any type of, maybe they need clotting factors or different proteins, maybe antibodies. And then platelets, pooled platelets. So they actually will take, there's a little layer, the white blood cells get thrown away and the platelet layer gets sucked off and gets pooled together with, I don't know, like 10 or 15 or so different donors. And then you get a pooled platelet that can be donated. And again, these are going to be less antigenic because they don't have a lot of features, a lot of antigens on their surface, but they can be somewhat antigenic. But red blood cells is usually what we're talking about when we talk about a blood transfusion. A quick little refresher, you might be familiar with what red blood cells look like antigenically, but we have really 4 major types of blood, A, B, and then O. And it has, they're named because of the surface antigen that they have. And the picture I have over here, this kind of picture here is a schematic. It's something I just put together to kind of show you, give you an idea that type A and type B have different types of antigens that look very differently. And then type AB people will have both of those antigens. Type O won't have either of those antigens. But in real life, the image over here on the right is probably a better depiction, because even those O blood groups still have an antigen on the surface. It just doesn't have one of these extra sugars on the end. And so the A antigen has a GalNAc attached at the end of this string of sugars. And then B antigen just has the gal attached at the end of the string. And so we don't see that on the O antigen. So there's still antigen on the O surface. It just doesn't have that extra sugar on the end. So then individuals that have, oops, did I miss a slide? No, okay. Then individuals that have type A blood will inherently have antibody against type B. Type B will have antibody against type A. But because type AB people have both A and B antigens on their surface, they won't have any antibody against the ABO blood groups because they have those antigens, and so that's known as self. And then type O will have both anti-A and anti-B. Now, please note that these antibodies, I have them kind of drawn as IgG, but in a real situation, individuals have these antibodies inherently against the other type of blood group antigen because of commensal bacteria having sugars that look like ABL blood groups. They wouldn't be type IgG, they'd be type IgM if somebody has never been exposed to the other type of blood, because IgM is produced first, and IgG would only come around if there was exposure and T cells were engaged to get B cells to switch isotypes. So just know that inherent antibodies are going to be IgM against the ABO blood groups. Now, the antigens then that differ between genetically unrelated members of a species, so from human to human, those are going to be called alloantigens. And we might introduce that on that first slide. And then this was that word that I said, alloantibodies. If somebody is exposed to an antigen that is different, or an alloantigen, then they can mount alloantibodies, different, non-self antibodies is essentially what that means. So let's look at what happens then if somebody is exposed to red blood cells that have alloantigens on them. So if it of these red blood cells, if they so they would sorry, look here, if a type O person is mistakenly transfused with group A or B, so we can pretend there's A or B on here, right? Then they will be those red blood cells will be attacked, or the antibodies will recognize, let's say there's let's say there's A, and those antibodies are going to recognize the A antigens on the red blood cells and join together, and we'll have what we call agglutination. Do I have that word up here? Agglutination. And this is what happens during a red blood cell transfusion that is incompatible of the red blood cells. So these antibodies will bind to the surface of the red blood cells and then end up joining multiple red blood cells together and cause this clumping. The clumping can end up inside vessels, especially like the kidney or other organs, cause major clumping and drastic events, but then complement is also activated, and then complement destruction of the red blood cells can occur. We can also have macrophages recognize the FC portion of the antibodies and take these red blood cells up and phagocytize them. So in the end, if an incompatible red blood cell is given to a recipient, they will destroy those red blood cells in some way or another. So then we end up having histocompatibility, or we probably don't call them histocompatible, we call them blood group compatible, or ABO compatible is probably the better term that is used, where type A people who have A on their red blood cells, they can donate to other A individuals, but also B also have A on their surface. Likewise, type B individuals who have B antigen on their surface can donate to other B individuals, but also AB. Now, type AB individuals, because they have both A and B antigens on their surface, are really only able to donate to other AB individuals. They're very limited in who they can donate to, where type O individuals are called the universal donor because they can donate to any of the blood groups because they don't have A or B on their surface. But let's throw in the fact that ABO is not the only antigen on the surface of red blood cells. Another major one is the Rh factor. So we say if somebody's A pos or O neg, right? We're referring to the Rh factor if they're negative or positive for the Rh antigen. Rh stands for rhesus. Anyway, it's the antigen that is the other big one that we are most concerned about when looking at blood transfusions. And then that adds another layer where O negative individuals don't have any A or B, nor do they have any Rh antigens. So they truly are the universal donor and can donate to any of the other blood groups. And then positive individuals for Rh can only donate to other positive individuals, and negative individuals can donate to any of them. But then we still have that, we still have this situation where we have to worry about ABO and Rh. So you can see that green means, yep, good to go. That being said, ABO, regardless, even if you are a type O donor, you are really probably only going to give to another O individual. If you're type A, you're going to give really only to type A. ABO is matched as best as they can. Now, ABO and Rh are two of the big ones that we're most concerned about because they tend to be the most antigenic. But there are about 30 other blood group antigens that are known and that are tested for, but not with the stringent first line testing that ABO and RH has done. So when you are ordering a transfusion, a blood transfusion for somebody, you actually order what's called a type and crossmatch, or a screen and crossmatch. And the type or the screening, that's looking for ABO and RH factor. And then what we do in the lab is we do a backwards test. So when we find somebody, we know their ABO profile, and we know their RH profile, we then go to the fridge, we pull out a blood sample. If it's A pos, we pull out an A pos, and we test. And in the test tube, we actually mix the recipient's blood with, the donor's blood with the recipient's plasma to see if there's agglutination, to see if there's any possibility that the recipient's immune system would attack. And so that's called a cross-match test. And this is going to look for any possible antibodies against those other ABO or those other blood group antigens. So it's kind of a backwards testing. And then if there's no agglutination, then we say, okay, we're pretty certain that the recipient will be able to tolerate the red blood cells that are given. So the underlying immunological mechanism provoked by ABO incompatibility is very similar to what we saw with a type 2 hypersensitivity. Recall that a type 2 hypersensitivity is antibody mediated, and it's when an antibody recognizes an altered self-protein. Now, that's not quite what happens with ABO incompatibility. Instead of having an altered self-antigen, we end up having just a totally different antigen on the surface, which is non-self, but it's still antibody-driven, and we end up having antibody attached to this would be like the donor red blood cell. And this would be like if this was type A and then this was anti-A, it would recognize, and we would have this FC portion recognized by a macrophage, and then this donor red blood cell would be either wiped out by complement activation or by phagocytosis, or we can also have the agglutination and destruction there. And so hemolytic reactions, we call them transfusion reactions, typically can look pretty mild, from like having a fever and chills to very, very severe, where shock can occur, renal failure, and then potentially death. But usually they can be stopped pretty quickly if any of these. So when somebody's getting a transfusion, they have a temperature monitor on them the whole time, and looking for any type of this type 2 hypersensitivity that you would see. So that is transfusions. We'll look at the other two big types of transplants in the next lecture.